Oh, hey there! Welcome to the nucleus, the core of life. Today, we are diving deeper into the topic of nucleic acids, the building blocks of DNA and RNA. More specifically, we'll be going over the structures of DNA and RNA with emphasis on the primary structure, its importance, features, the nucleotide sequence, and importance and structure. By the end of this video, we'd like you guys to be able to describe the nucleic acids, identify the parts of the primary structure of DNA and RNA, illustrate the primary structure of DNA and RNA given a sequence, and reflect on the importance of the primary structure of DNA and RNA. Buckle up for a wild ride as we unravel the secrets of the primary structure. It's gonna be nucleic! Ever wondered what makes you, you? The answer lies in the intricate world of nucleic acids, the building blocks of life. These incredible molecules hold the blueprint for every organism on Earth. But how do they do it? Let's break it down, start with its very foundation, the primary structure. But what exactly are nucleic acids? Nucleic acids are made of chains of monomers called nucleotides. These chains have structures that vary in complexity, including primary, secondary, and higher structures. Today, we'll focus on the foundation, the primary structure. Imagine these nucleotides as the alphabet of life. They link together to form chains, much like letters forming words. The order of these nucleotides are crucial since they dictate the synthesis of proteins that eventually make up you and me. Stay tuned as we unravel the mysteries: adenine, guanine, cytosine, thymine, and uracil, the 5 letters that spell out the story of life. Starting with the structures of DNA and RNA. These molecules are like the secret codes of life, but in order for us to crack the code, we have to start with the basics. Take a look at these structures. You'll notice that the composition of DNA and RNA are essentially similar. However, DNA is double-stranded in a helix formation, while RNA is single-stranded. Now, imagine we unzip the DNA strand like a zipper. You'll see that it has a distinct beginning and end, exhibiting a clear directionality. One end is designated as the 5' end, while the other end is the 3' end. This directionality is due to the phosphodiester bonds linking the sugar molecules in the backbone, which connect the 3' carbon of one sugar to the 5' carbon of the next. At the ends of each DNA or RNA strand, you'll usually find these certain groups. One end would have an uncreated phosphate group, while the other end would have an unreacted hydroxyl group. It is at these points where more nucleic acids can connect, further building and modifying the DNA or RNA strand. So we've established that DNA and RNA have directionality. But what else sets these molecules apart? Remember those nucleotides we've talked about? Adenine, thymine, cytosine, and guanine make up DNA, with thymine being replaced by uracil and RNA. Well, the order of these bases is what gives each nucleic acid its unique identity. It's like a genetic code written in 4 letters. This order of bases is called the nucleotide sequence. It's the foundation for the incredible diversity of life on Earth. From your neighbor's dog to us humans, the unique sequence of nucleotides in our DNA is what makes us different despite once coming from a single fertilized cell. And so, it is in the highly specific nucleotide sequence that we can say DNA and RNA not only have directionality, but they also have individuality. Okay, we've seen how nucleotides link up to form DNA or RNA strands. But how do we efficiently represent this complex structure? If I were to draw it out every single time I want to show DNA or RNA, I'd just be wasting tons of paper and everybody's time. So that's why scientists develop several shorthand notations to make our lives easier. Let's work with an example to illustrate these. So here we have a detailed nucleotide sequence containing thymine, guanine, cytosine, and adenine. Since thymine is present and not uracil, this is a DNA structure. We can also see that the 5' end has a phosphate group, and the 3' end has a hydroxyl group. Check out this notation. We'll start by writing a P representing the phosphate group and drawing a line downwards to the carbon it's attached to. We'll note that this is the 5' end. Next, we draw a straight line upwards and write T, since thymine is the first nitrogenous base in this sequence. We'll draw a short line downward and note 3' to represent the carbon that the hydroxyl group is at. However, we don't write the OH yet since we aren't done with this sequence. So, we simply repeat this pattern for every other base. We'll use P to denote the phosphate groups and draw lines to represent where they connect, with the respective bases noted by their letter form. And, at the very end, we make sure that denote the unreacted hydroxyl group. With this diagram, we can represent short DNA sequences. The letter T, G, C, and A stand for the 4 nucleotide bases: thymine, guanine, cytosine, and adenine. The P represents the phosphate group, and the 5' and 3' numbers indicate the directionality of the strand. So this compact notation captures the essential information about the DNA sequence without cluttering the diagram. But is there a way to write this down without showing the structure? The answer is yes. Now, if we assume that all of the phosphodiester links connect to a 5' phosphate to a 3' hydroxyl, which is usually the case, we can use a more compact notation for our nucleotide sequences. Instead of writing out all the details, we can simplify things by writing it as PT, PG, PC, PA. Cool, right? This makes it way more efficient and easier to read. Note that we don't have to write down the hydroxyl groups, since it's understood that these are either a part of the phosphodiester bonds that hold the sequence together or simply unreacted. But what if the situation was flipped? If the phosphate is at the 3' end and the hydroxyl at the 5' end, then we would simply write it as TP, GP, CP, AP to show that the phosphate group is no longer at the 5' end, but rather at the 3' end. See how just the placement of P dictates the direction of the strand? The difference, whether it is written before or after the nucleotide base, is really important. So always remember to pay attention to where the phosphate group is located. And finally, if we're only concerned with the sequence of bases in the structure, which, let's face it, is often the case, we can simplify it even further. We can write it in an ultra-compact form as TGCA. This notation strips it down to the essentials, making it super easy to read and work with. The only downside is that we won't know the direction of the strand unless it's noted down. But hey, simplicity is key. So we've learned about the primary structure of nucleic acids and how to identify and write them down. But what's so important about this particular structure? Are they not just the most basic, broken down version of what makes us up? The primary importance of the primary structure, or sequence, lies in its pivotal role as the genetic blueprint stored within DNA. Think of a gene as a unique DNA sequence, encoded in a 4-letter language where each letter represents a base. These sequences are absolutely crucial because they are the building blocks that allow our bodies to synthesize proteins. These proteins, in turn, kickstart a cascade of vital processes that ultimately define who we are, from the inside and out. Alright folks, that concludes our discussion. Now it's time to take a moment and recap what we've covered and see if you can remember what we covered in this video. You'll be given 15 seconds to think about your answer before you reveal it to you. But if you need more time, feel free to pause this video. Ready? Let's start. What are the monomers that make up nucleic acids? That's right, it's nucleotides. Now, what specific structure of nucleotides did we talk about today? Two for two is the primary structure. How about what are the bases found in DNA and RNA? Correct! In DNA, we find TGCA, whereas in RNA, it's UGCA. And last but not the least, what is the name of this structure? Note to include the phosphodiester links. Is this structure a DNA or RNA? You're absolutely nailing it! It's PCGPAPU, and it's an RNA because it contains U. Alright, that wraps it up for now, everyone. I hope today's exploration of DNA and RNA gave you plenty to think about. Until next time, take care and see you in our next video.